Good morning, children. Good morning, children. The topic name today is three domains of life. Yesterday we discussed up to this chapter on this topic only. So we are going to discuss complete details about three domains. Okay. Number one, domain archaea. What are the three domains? Archaea, bacteria, and eukarya. So here domain archaea I am giving information. Domain archaea. Okay. So what is this archaea? That is important. First one, single-celled organisms. In this domain, they included single-celled organisms. First point. Number two, which have the extreme conditions. These these organisms have ability to survive in extreme conditions. Very, very important, the word extreme conditions. Okay, extreme conditions. Like, what are the extreme conditions? Like volcano vents, number one, and hot springs, number two, hot springs, and polar ice caps, and polar ice caps. Remember these names. And that's why extreme condition they are living, no, that's why they will be called extremophiles. What is the meaning of phili? Means loving. So they love extreme conditions. Phili means what? Loving. So they can live in extreme conditions. So next important, they are capable of, they can synthesize, very, very important, their food without sunlight. They can synthesize the food. Synthesize their food without sunlight. Without sunlight, they can synthesize. Oh, these extremophiles. Okay, and also without sunlight and oxygen. Sunlight and oxygen, they can synthesize the food. How? By utilizing very, very important, hydrogen sulfide. By utilizing hydrogen sulfide and other chemicals. Next important point, and other chemicals. These points you should have to remember. Next one, from where? In the which one? Especially in volcanic vents. You know, volcano means volcanoes, how much temperature will be there. And some methanogens which produce the methane, methanogenic bacteria. Next one, another important one, methane. Okay, another important one, you know, some salty conditions, they will be called halophiles, and thermoacidophiles. So acidic environments and high temperatures. Acidic environment and high temperatures, they can survive. So this is about domain archaea. Come to next domain, what I said, domain bacteria. You know very well about bacteria. Why? It's a prokaryotes. First point, whenever you hear the word bacteria, first you have to remember they are prokaryotic. No, what is the prokaryote means? No definite nucleus. No definite nucleus. Next, and DNA exists DNA only exists as a DNA exists as a circular chromosomes. Circular chromosomes, very important point in the prokaryotes. Okay, and no histone proteins. Histone proteins means which is fix the DNA. Histone proteins also, histone proteins are absent. Next, no membrane bound organelles. You know cell organelles, mitochondria, Golgi complex, like that. Membrane, no membrane bound organelles in the cells in this prokaryotic cell bacteria. Only then what is there? What is there in the sense 70S type. There are two types of ribosomes, 80S and 70S. Here 70S ribosomes are present. Next, the cell wall contains very important peptidoglycan. Very glycan. Peptidoglycan is present. Okay, peptidoglycans. Next one, all are decomposers. Many are decomposers, means which decompose the organic matters. Okay. Next, so photosynthesizers, some are only some photosynthesizers. Photosynthesize means which can involve in the photosynthesis process. Okay, some are photosynthesizers. What you can say, photosynthesizers. This one you should have to remember. And these are causes. Some are causes diseases. Some microbes, some bacteria causes diseases. Some are useful. Okay, so there by beneficial means beneficial also there. The beneficial bacteria are called, what do you call the beneficial bacteria are called? Probiotic. What do you call? Probiotic. If harmful means they will be called harmful bacteria, in the sense they will be called pathogenic bacteria. Okay, pathogenic bacteria. And the next one, very important, cyanobacteria, which can involve in the photosynthesis. We can involve in the photosynthesis. Okay, so like this, different concepts are there in the bacteria. Okay, once again we are going to discuss what we are there. What are the three domain concepts? We know Carl Woese. Carl Woese is a scientist, first developed the domain concept. Domain is higher than the kingdom. The domain will give complete picture about the living organisms. So here three types of domains are there. Carl Woese divided the three domains system. So three domains are, number one, domain archy. Domain archy contains, first one, what they are, single celled organisms, means only one cell, that's it. The ability, these archaebacteria have ability, they have special capacity to survive in extreme conditions. Extreme conditions in the sense very high temperatures or very low temperatures or very high acidic conditions or salty conditions. Such kind of conditions are called extreme conditions, like volcano vents. You know how much temperature is there in volcanoes. Hot springs, volcano vents, hot springs, and polar ice caps. Extremophils. What is a mean? Phil means what? What is a philophiles? Philly means what? You know, loving. They live in the extreme conditions. That's why they are extreme conditions loving. No, that's why these archaebacteria or archae-related, domain-related organisms are called, what do you call? Extremophils. Next, they synthesize the food. How these archaebacteria can synthesize the food? Without sunlight, very important point, and oxygen. They don't require the sunlight and oxygen. But what they require then? By utilizing hydrogen sulfate, very important point. They don't want any sunlight and oxygen, but they depend on hydrogen sulfide for food. And not only hydrogen sulfide, and other chemicals. What are the other chemicals? Like sulfur, like nitrogen, like so phosphorus, like that other chemicals, they will depend and they will produce the food. Especially the hydrogen sulfide and other chemicals using extremophiles live in which area? In the volcanic vents or volcanic vents. Next, some more extremophiles examples, methanogens. You know, methanogens, extreme conditions only these are present, and they producing the methane, you know, biogas. That's why they are called methanogens, produce the methane. And some extremophiles, high salty, you believe or not, even high salty conditions also, some microbes, especially these archaea related bacteria, survive. Those are called halophiles. So next, some can survive in acidic environment with high temperature. They will be called thermoacidophiles. What do you called? They will be called thermoacidophiles. Next one, domain bacteria. So these all are the characters of what you called archaea. The second domain. What is the second domain? The second domain is prokaryotic bacteria. Simply you can say second domain is bacteria. So bacteria you are studying from smallhood onwards, but we have to give little clarity. Bacteria is the prokaryotes. In the name itself, answer is there. Pro, no, or first, undeveloped. Karyote, nucleus. So no definite nucleus. So in the bacteria, no definite nucleus. But DNA as a circular chromosomes, and there will be DNA is present, but DNA is present, nucleus is absent, but DNA is present. And where it is present? As a or in a circular chromosomes they exist. DNA exists. How DNA exists? As a in a circular chromosomes. And DNA don't connect with the are fixed by the help of histones. Why? The histone proteins are very important. Histone proteins are absent. See, very long DNA should have to fix in the small cell. In the sense, compulsory histone proteins are required. But in this case, in the prokaryotic cells, the histone proteins are absent. And no membrane bound organelles, you know, mitochondria, Golgi complex, and say what you call such kind of endoplasmic reticulum, that kind of all organelles are absent. Then what are there? Which type of organelles are there? Only 70S type. 70S. What is S? S is Svedberg unit. So 70S type of ribosomes are present. In the bacterial cell wall, you know, peptidoglycans. These are also some important components of the cell wall. So peptidoglycans are present. Next one, the cell wall, peptidoglycans, NAM, NAG, N-acetyl muramic acid, N-acetyl glutamic acid, like that materials are there. Next, some of the bacteria are decomposers. What is the decomposers? Which will be decompose the dead materials, dead organic materials. Okay, and some are photosynthesizers, means they can prepare, involve in the photosynthesis process to produce the food and to produce oxygen. They are example of photosynthetic bacteria, cyanobacteria. Some causes diseases. So some are beneficial bacteria. You know very well. Some, suppose cholera, Vibrio cholerae is causes the disease that is related to bacteria. Some are beneficial, and suppose we want to convert the milk to curd. You know very well which bacteria? Lactobacillus. Same like that. Which are useful? Those bacteria are called probiotic. Harmful bacteria is there in the sense we are calling, they will be called pathogenic. Like that, the complete information is there in our syllabus about what? Bacteria and archaea. Bacteria and archaea. So next domain is, one more domain is there, that is called eukarya. What do you call the next domain? Eukarya. Next domain is what is called eukarya. So domain eukarya. Domain eukarya. So domain eukarya. What is this? Eukaryote animals. All eukaryotic animals which are coming under this domain. Eukaryotic means they have, first word is main eukaryote means they have true nucleus. Okay, and also second point what you have to remember, they have membrane bound organelles. They have means mitochondria, Golgi complex. Okay, these are present. Next, DNA is nuclear. DNA is there. That is linear chromosomes. Linear means you know very well. In the linear chromosomes, DNA is present. This point also you should have to arrange it in linear chromosomes. And most what are there not there in the prokaryotes, here all present. So with the help of how they will arrange in the linear chromosomes, by the help of histone proteins. So next one is ribosomes is there. Ribosomes. What type of ribosomes are there in this? You know, type of ribosomes are ATS. Okay, and also in the cytosol, cytoplasm, 70S type, and so ribosomes are 80S type in the cytosol and 70S type. Okay, 80S type is there in the cytosol and 70S type also there in the chloroplast, and very important bit here. Okay, chloroplast and mitochondria having 70S ribosomes. Okay, so next namely these domain under so many kingdoms are there. What are the kingdoms? You know very well. Protista, fungi, okay, and plante, and animalia. So like this the kingdoms also there. So now in 1987, very important, in 1987 the scientist name, please remember this name, Cavalier Smith revised the six kingdom. There are so many kingdoms are there here. Six kingdom system to seven kingdom system. I will give complete picture about these kingdoms, but you should remember now. So the scientist who revised the six kingdom concept to seven kingdom concept. Okay, kingdom was introduced, kingdom system. Okay, kingdom introduced and revised. Okay, so this one, which one? Super kingdom introduced and revised it to seven kingdom. Okay, revised it to seven kingdom concept. Okay, so first one, what? Two kingdom concept. Okay, first one is two kingdom concept. Only what are the two kingdoms? Prokaryotes and eukaryotes. Okay, next what are those? So here seven kingdoms. In that seven kingdoms, what you have to do? First, what I'm telling is 1987, Cavalier and Smith revised the six kingdom system to seven kingdom. Why? The super kingdom they introduced and revised into seven kingdoms. So what are that one? Generally, you know, Monera, Protista, fungi, plante, animalia. In that, Monera is again they divide into prokaryotes and eukaryotes. And here one more is there. What are those? What are those kingdoms? Come on, here I'm giving first three kingdom concept. Three kingdom concept means Monera, Protista, sorry, five kingdom: Monera, Protista, fungi, Plantae, Animalia. These all are called five kingdom concepts. The sixth kingdom concept is sixth kingdom. Additionally, what will be there? So bacteria. Observe very careful. Bacteria, archaea, and same Protista, fungi, Plantae, Animalia. Okay, Protista, plants. Next one, seven kingdom. What are that one? Eubacteria and Archaebacteria as it is. But here one more is there. Protozoa. Next, Chromista, fungi, Plantae, and you know very well what are those? Animalia. Okay, so first what we have to remember is the kingdom concepts are there. What are the kingdom concepts? Two kingdom concept, three kingdom concept are there. So seven kingdom concepts. Okay, five kingdom concepts also given because of these all animals. These are microorganisms. Okay, so first one, domain Eukarya. Eukaryotic animals. They contains what? True nucleus. Then they are membrane bound organelles, as it is general about our cells like that. DNA will be linear. Chromosomes, in linear chromosomes DNA is present with connected with histone proteins. Ribosomes are ATS type. They will be in the cytosol of the cell. Even the eukaryotes have 70S type ribosomes. They are in the chloroplast and mitochondria. There are different types of kingdoms proposed, and these all Protista to Animalia. All are eukaryotes, Protista, fungi, Plantae, Animalia. All are what is that? Eukaryotes. Okay, comes under the eukaryotes. Later, there are so many kingdoms, but here clarity. 1987, the two scientists. Who are the two scientists? Remember, Needbit, Cavalier, and Smith revised the six kingdom. Up to that six kingdom system to seven kingdom. Why the seven kingdom? Why to seven kingdoms are the Eubacteria is there, Archaea is there, Protozoa is there. Additionally, what they are there? Added Chromista. What is Chromista? Based upon pigments, based upon presence of color giving pigments. Those organisms are separated and kept under separate kingdom. So the two kingdoms, the bacteria, Monera, divide into prokaryotes and eukaryotes like that. Okay, so first five kingdom, Whittaker is the scientist. Okay, who is the scientist? R.H. Whittaker. Remember this name. R.H. Whittaker proposed the five kingdom: Monera, Protista, fungi, Plantae, Animalia. Then six kingdom. What are those? Monera divide into bacteria and archaea. So bacteria kingdom, archaea, and Protozoa, fungi, Plantae, Animalia. Count it: 1, 2, 3, 4, 5, 6. Six kingdom concept. Later, seven kingdom concept. This is six. The seven kingdom concept. What are those? Eubacteria, archaea, Protozoa, Chromista, fungi, Plantae, Animalia. So based upon the color, this additional kingdom came, that is called Chromista. So this is about what we called the kingdom systems. Okay. The next one, you know very well, the taxonomic hierarchy. We are going to discuss about that one. Taxonomic hierarchy. What is taxonomic hierarchy? What are the complete species, genus, family, order, class, phylum, kingdom? We are going to discuss. Okay, so first we have to go for that one. Okay, there will be in the textbook, if you observe, there will be chart is there. That one you have to read about all the kingdoms. Okay, next, taxonomic what do you call taxonomic hierarchy. Okay, taxonomic hierarchy. What is this one? You know very well. There is hierarchy includes 7 major categories. How many categories? 7 major categories. What are the 7 major categories? You know very well. Come on, write. Tell. Species, genus, family, order, class, phylum, kingdom. 1, 2, 3, 4, 5, 6, 7. What is species, genus, family, order, class, phylum, and what do you call kingdom? These are the hierarchy. Later, so other intermediate categories also there. Other intermediate categories. Other intermediate. But these are not that much, but anyhow useful. Intermediate categories are there. So what are the other intermediate categories? Number 1, sub-kingdom. So kingdom after sub-kingdom, and grade, then division. Okay, division, subdivision, division, subdivision, subphylum, next superclass, then subclass. Okay, superclass, subclass, super order. Okay, super order, suborder. Okay, like this. Then super family. Okay, super family. Then subfamily. Okay, subfamily. Then subspecies. Like this. Some are there. The intermediate categories. What are those? Subkingdom, grade, division, subdivision, subphylum, superclass, subclass, superorder, suborder, superfamily, subfamily, subspecies. Like that, the intermediate categories also there. But major categories what? The major categories are these: species, genus, family, order, class, phylum, kingdom in the taxonomic hierarchies. Okay, now one by one we will be discuss. Okay, now what we have to do? One by one we have to discuss. Number 1, species. What is that one? Species. Species means what? It's a basic, very very important word. Species, basic unit of classification. What is the species? Basic unit of classification. Okay, is a group of animals, group of animals having similar, very very important, having similar morphological features. Okay, morphological features, and is reproductively isolated. Okay, reproductively isolated to produce offspring. This is a very important point, to produce offspring. So this is the one of the important. So for example, monkey crossed with monkey, then only the fertile offspring will be come. This is all togetherly considered as a particular species. But some certain what we call exceptional animals, some exceptional animals are there. Exceptional animals. What are the exceptional animals? Sterile offsprings. Some exceptional animals produce sterile. Sterile means they can't give birth. So can produce sterile, okay, offsprings. What are the best examples? Sterile offsprings. Come on, can you repeat that? Hinnies, mules, okay, hinnies and mules. Suppose male horse, okay, male horse and female donkey. Male horse and female donkey, in the sense, you know very well what we'll get. The result is hinni. It's sterile. The next one, male donkey and female horse crossed, in the sense, female horse crossed, in the sense, you know very well, one will become mules. These are also sterile. Then male lion, okay, and female tiger, so you know liger will come, very very important bits, and male tiger and female lion crossed, in the sense, you know very well what you will get, the tigon. What is that? Tigon. So these points you should have to remember. What are the points you should remember? Also observe carefully once again. So taxonomic hierarchy contains 7 major categories. What are the 7 major categories in the taxonomic categories? Species, genus, family, order, class, phylum, kingdom. These are the first. Then what are the other intermediate categories also there? The other intermediate categories, what is the first one? Sub-kingdom, then grade, then division, then subdivision, then subphylum, then superclass, then subclass, then superorder, then suborder, then superfamily, then subfamily, then subspecies. Like that also is there. But very rarely we will use this. Only some cases. Here division means, you know very well, instead of in the plant kingdoms, the division is replaced, phylum is replaced with division. That point you should have to remember. Next come to species. This is a neat bit. Division for what? Phylum in the animal kingdom, division in the plant kingdom, we'll say like that. Next, species. Basic unit of classification. What is the species definition? Compulsory basic first unit of classification. How to define the species? In the sense, group of animals having similar morphological features and is reproductively isolated to produce offspring. Suppose monkey is the example I given. Monkey characters and monkey, you know very well, similar characters. These all similar characters, all animals are called monkeys. And when they crossed, again what should have to come? Monkey. Means these all characteristic animals all are particularly, they will be called species, monkey species, like that we can say. So but some cases, monkey crossed with monkey, in the sense monkey will come, but some cases, some exceptional animals produce sterile offsprings. What are those? Male horse crossed with female donkey, in the sense, will get inni. Male donkey crossed with female horse, in the sense, mule. Male lion, female tiger means liger. Male tiger, female lion crossed, in the sense, we will get tigon. So these all the things will be there. In the next class, we will be discuss about what is genus, what is family, what is order, what is class, what is phylum, what is kingdom will be discussed. Okay. Thank you children. Next class remaining categories we will discuss.